Yeah, that was helpful in your email. Rhymes with jelly.
Yep. I thought about putting it in my email signature.
Oh, why not? Then you wouldn't get, I mean, I don't know. It depends how much it annoys you, right?
Yeah, I mean, it doesn't. Val has happened and it's fine.
Right, looks like we are good live on YouTube, so we can go ahead and get started whenever folks are ready. Sorry, just to double check, so we have each of you for sort of a half-hour slot, inclusive of questions, and we'll have Katrina go first because she's more junior.
Thanks.
Yeah, sorry, I was just tweeting that. Okay, so let's get started. Hi, everyone. We've got a double header speakers today. So our first speaker is Katrina Velli. She did her PhD in Ken Campoloni's lab at University of Connecticut, working on how and why pathogenic E. coli hijack the actin cytoskeleton of the cells they infect. And then in 2008, she joined the lab of Lillian Fritz-Lehlen at the University of Massachusetts Amherst as a postdoc, where she is studying actin-driven phenotypes in a model system for the brain-eating amoeba. So you can take it away. Thanks, Katrina.
All right, thanks for that introduction, and thanks to the organizers for giving me the opportunity to present some of my work here today in this cell migration seminar series. So today I'm going to be talking about Naegleria grouperi, which are these cells you see in the background here. I'll turn on the laser pointer. And I've been studying them for about 3 years in Lil Fritz-Lehlen's lab. And if you've never heard of Naegleria, there are a few key reasons why you might want to be studying them. So one reason is that one species of Naegleria, Naegleria fowleri, which I'll point out is not the one I'm studying, but Naegleria fowleri is also known as the brain-eating amoeba. So N. fowleri thrives in warm freshwater environments, so people typically get infected if they go swimming in a pond on a hot summer day and they get water up their nose. The amoebae can then crawl along your olfactory bulb and into your brain, where they cause a massive inflammatory response and also start to literally digest pieces of brain tissue. And unfortunately, there are no reliable treatments for this disease, resulting in over a 90% case fatality rate. So in order to come up with some new, more effective treatments, we really need to understand the basic cell biology of Naegleria. The second reason you might want to study Naegleria comes from an evolutionary perspective. So on this simplified eukaryotic tree of life, you can see Naegleria up here within the Discoba lineage. And this is also where you find things like trypanosomes and euglena. But most of the cell biology that we have detailed mechanisms for has come from this episthecont lineage, which encompasses yeast to human scale diversity and most of the model organisms that we know of. And because Naegleria in the Discoba lineage diverged from the episthecont lineage about 1 or 2 billion years ago, it's really hard to assume that any basic cell biology has been conserved. Anything that is conserved between Naegleria and epistheconts can give us clues as to what the last common eukaryotic ancestor might have looked like. And a third reason you might want to study Naegleria, and the main reason our lab is so interested in Naegleria, is because of its really unique and amazing cytoskeletal system. So as an interphase amoeba cell, these cells have no microtubule cytoskeleton at all. And I can't emphasize enough how crazy that is. Out of all the eukaryotic species that we know of, this and one other amoeba have been described as having no interphase microtubules. Now, I say interphase because these amoebae do still use a mitotic spindle made of tubulin to divide, but it's a closed mitosis, so the nuclear envelope doesn't break down. And following cell division, the cells get rid of all of their tubulin and tubulin transcripts and revert back to this microtubule-free amoeboid state. Now, this spindle is still pretty interesting, and it's built with some divergent tubulins. So if you're interested in that, we do have a recent preprint on bioRxiv about those spindles. But Naegleria can also use tubulin for something else. So Naegleria can transiently differentiate into this flagellated type cell. So to do that, it needs to transcribe, translate, and build an entire microtubule cytoskeleton from scratch. And it can do that in about an hour, which is a really amazing cellular feat. And after minutes to hours of swimming around as a flagellate, it will revert back to being an amoeba, degrading all of the tubulin and tubulin transcripts. And while this has been used to study the assembly of microtubule systems, no one had really leveraged this system to study actin without any microtubule crosstalk. So I've been interested in looking at actin in these amoebae, and some of what I'm going to tell you today has already been published in JCB. So speaking of actin, our lab really loves actin, and organisms from all across the tree of life are using actin to build a wide variety of different structures. So we have mammalian cells over here that can build stress fibers and thin lamellipodial projections. And we also have fission yeast over here with their cytokinetic rings. And it's not just eukaryotes that are using actin. In fact, you can see Caulobacter crescentus up here with its homolog MREB. And in fact, eukaryotic actin is thought to have evolved from an archaeal actin. So really, every domain of life is using actin for something. And as I'm sure most of you already know, actin alone is not great at forming these structures. So you need actin nucleators. And most cells encode an ARP23 complex, which can, when activated, bind to the side of a pre-existing actin filament to nucleate a new filament off the side. And this is really great at forming branched actin networks that push on cell membranes, particularly at the leading edge of the cell. Most cells also encode formin family proteins, which can stabilize these actin dimers to nucleate actin, as well as elongate actin filaments. And from the Naegleria genome, we know that Naegleria has about 30 actins, as well as a full ARP23 complex with 2 ARP2 subunits, as well as the ARP23 activators, WASP, scar wave complex, and WASH complex. Naegleria also encodes 14 formin family proteins, including 9 diaphanous-like formins and 3 formins that look typical of plants. So Naegleria has a lot in its actin toolkit. So my question is what Naegleria is using all this actin for? And in particular, I want to know how Naegleria amoebae are crawling and how they're dealing with osmotic stress. And osmoregulation might not be your first thought when you think of actin phenotypes, but if that's your only cytoskeletal system and you're a freshwater protist, you really need to be able to deal with water rushing into your cell. So I'll talk about that for the last few minutes of my talk, because first I want to talk about how these cells are crawling. So if we just watch them crawl on a glass cover slip, you can see they have this flat lamellar protrusion at the front of the cell. And I just want to mention that while this is sped up, it's not sped up very much. These cells crawl incredibly fast. So this is not the really slow creeping motility that's a few microns an hour that highly adherent cells with focal adhesions use. So they're moving quite fast with this lamellar ruffle at the front. And if we were to just look at it, it looks a lot like the actin ruffles we see in other cell types, like this mammalian leukocyte. You can see it has this lamellar ruffle as it's crawling through this collagen matrix. But we can't just assume that there's actin in here because there's actin in there. And a great example of this comes from nematode sperm, which phenotypically, you can see this pseudopod in retrograde flow in this pseudopod. And you could think it's actin, but it's not. It's actually a different filament system entirely called major sperm protein. So we need to find out if there's actually actin in those protrusions. So if we take cells and fix them and stain them for actin, or polymerized actin, we can see they have these F-actin-rich lamellar ruffles. And if we follow that up with scanning electron microscopy, we can confirm that these are very thin ruffles. So if this was like a mammalian cell, you could imagine ARP23 complex is what's responsible for polymerizing actin in those ruffles. So to test this, I use small molecule inhibitors. So I use latrunculin to prevent all actin assembly, CK666 to inhibit the ARP23 complex, and SMIF2 to broadly inhibit Naegleria's 14 formins. And I also want to note the cool work that just came out of Jim Seller's lab, showing that SMIF2 can also inhibit myosins. So we've been thinking about this as kind of a broad inhibitor for contractile actin networks. So when we want to look at actin ARP23 in terms of motility and making these ruffles, we can do the same experiments I just showed you. So we have our control cells stained for actin, which have lots of ruffles. If we inhibit actin assembly with latrunculin, the cells round up and they lose all their ruffles. And if we treat cells with the ARP2/3 complex inhibitors, the cells lose their ruffles, but instead they make these spikes. And this is pretty cool because we know from mammalian systems that when you inhibit the ARP2/3 complex, those cells will make spikes preferentially, or phylopodia preferentially. And if you remember back to Jim Baer's seminar a couple weeks ago, he showed his ARPC2 knockout cells, which were making phylopodia instead of lamellipodia. So something similar could be happening here. So what happens if we look at these cells live? So here are some cells. They're going to be crawling in buffer first, and then I'm adding the ARP2/3 complex inhibitor CK666. So if you follow this cell, you can see it crawls fine before treatment. And then upon treatment there, all of the cells are either stopping all movement or they're slowing down significantly. So we can quantify that across dozens of cells. And here I just want to plug the idea of superplots. So each tiny dot on this plot represents the speed of one individual cell. And the larger symbols represent experimental level averages. So they're all coordinated by shape, so you can really embrace your cell-to-cell variability and also look at your run-to-run variability. And you can see that across all three trials. With latrunculin, if you can't assemble actin, you can't move. And with CK666 treatment, the cells are moving significantly slower, which is, again, what we would expect from mammalian cells. So if we look at this at a little higher resolution, if you follow this cell here, the cell is moving fine before treatment. You can see its nice lamellar protrusion at the front. But after treatment there, the cells slow down, they get blebby, and then they start to build their phylopodia. And now we can say that these structures that are growing are phylopodia, because if we fix and stain in one step, there you can see that they are indeed filled with actin polymer. So this means that Naegleria are using actin and ARP2/3 complex to move. So despite 1 or 2 billion years of independent evolution, Naegleria and mammalian cells are using the same mechanisms. And this also implies that the last common eukaryotic ancestor may have also used this mechanism to move. So now I want to talk a little bit about directionality, maintaining a front and a back. So here we have an experiment set up the same way. So cells are untreated at first, and then I'm going to be adding the formin inhibitor SMIF2. So if you follow this cell, you can see it's moving fine before treatment. I added SMIF2 there, and then the cells just start kind of moving in circles. They're not moving productively. So I measured this using directional persistence. So I took the displacement and divided that by the total distance as a reminder of that metric. So a value of 1 would be a perfectly direct route, and increasingly smaller values are more roundabout paths. So if we look at this for control cells compared to all of our drug treatments, you can see that treatment with the formin or contractile network inhibitor SMIF2 significantly reduces persistence. So those cells aren't maintaining their polarity or moving productively. So if you're interested in everything that I've just talked about, you can look at it in even more excruciating detail in our JCB paper. But now I want to shift gears a little bit and talk about some unpublished data about how these cells are dealing with osmotic stress. So I kind of stumbled upon this phenotype while I was doing the same experiments I've already shown you, but I was using water instead of buffer. So watch what happens when you have cells crawling in water and then you add SMIF2. All of the cells start exploding like popcorn. So at first I'm thinking, oh no, it's just an off-target effect of SMIF2. Maybe it's like messing with the cell membrane. So I took this data to my advisor, Lil, and she encouraged me to try to think about it as a defect in osmoregulation and see if I could rule that out. So let's take a step back and think about that for a minute. So this is no newsflash, I know, but freshwater protists live in fresh water. So they need to be constantly dealing with water rushing into their cells. So some cells use a cell wall to prevent this, and other cells use an organelle called the contractile vacuole. So you can see in this paramecium, it has this kind of array of tubes which collect water and empty into the central bladder, which pumps. So if you think about it, if you think about being maybe in a boat on this pond and you have a hole in your boat, you're going to be bailing out water constantly to prevent sinking. So just like that, these cells have to be constantly bailing out water to prevent their plasma membrane from rupturing. So thinking about it in this context, I wanted to see if I could rescue the SMIF2 phenotype by decreasing the amount of osmotic stress that the cells were under. So I've been working with an amazing undergrad in the lab, Tati, and what we've been doing is looking at cells that are either in water, which is a lot of hypoosmotic stress, or different concentrations of sorbitol, which is a non-metabolizable sugar that you can use to increase the osmolarity of the solution. So we looked at some cells that were control treated in gray here, and you can see that if you look at the first frame of the movie, and after an hour of treatment, there's still around the same % of cells in that field of view. And if we look at SMIF2 treatment, when the cells are in water, just like that first video I showed you, most of the cells, about 80% of the cells are bursting, but you can rescue this with increasing amounts of sorbitol. So less hypoosmotic stress rescues this phenotype, which means that this was in fact an osmoregulation defect that we see with SMIF2. So formins or contractile actin networks seem to be involved in contractile vacuole dynamics or in osmoregulation. So I wanted to dig a little deeper into this and find out what these contractile vacuoles look like. If you were to do a search for Naegleria contractile vacuoles, you wouldn't come up with much. So I wanted to compare to other protists as kind of an evolutionary perspective, and also to find out what they look like in other systems and how they're working there. So I've already shown you the contractile vacuoles in Paramecium, and these were actually discovered a ridiculously long time ago, actually in 1799 by Spallanzani, and he described this array of tubules that collects water into a bladder, and then the bladder pumps. And about 200 years later, we had our first marker for contractile vacuoles with a GFP tag. So this is GFP dagumen, and you can see it in green here, and you can see how extensive this contractile vacuole membrane network is, and you can even see a pumping event right there. About 20 years after that, John Hammer's lab found that for dictyostelium, contractile vacuole dynamics need actin, a myosin 5, and microtubules. And I'll just remind you at this point that Naegleria doesn't have microtubules, and it actually also doesn't have any myosin 5s. But this idea of actin and myosins being involved here seems likely given our SMIF2 result. So what did these structures look like in Naegleria? Well, if we look at them by phase contrast microscopy under agarose, you can see that there's a vacuole there that's forming from several smaller vacuoles that kind of merging their contents into one main bladder that's pumping. And if we look at that, what's happening with the membrane, we can use the membrane dye FM464. And you can see here that there's still a lot of, all that membrane is retained after the vacuole pumps. So if we look at this from still frames, you can see all the smaller vacuoles kind of merging their contents into eventually one larger vacuole. And after that pumps, all that membrane is retained. This seems similar to other systems where you have this tubular network of membrane in a main bladder. So next I wanted to look at what was happening with these vacuoles when they're responding to different osmotic pressures. So if you think back to that boat analogy I mentioned, if you have more hypoosmotic stress, say you have a couple more holes in your boat, then you need to do something about that, right? You either need to bail water out faster, increase the rate you're pumping water out, or you need a bigger bucket. You need to pump out more water with each push. So looking at these cells in water under more hypoosmotic stress, you can see they're forming fairly large contractile vacuoles. But if we look at them when they're under less hypoosmotic stress, they're in sorbitol, you can see these vacuoles are still pumping pretty regularly, but they're not anywhere near the size that they were when they were in water. So we quantified this and found that Naegleria is essentially changing the size of its bucket, right? So if it's in water, if it's under a lot of hypoosmotic stress, it's making these big vacuoles even when you normalize for cell area. Whereas when it's in increasing amounts of sorbitol, it's making smaller vacuoles, it's pumping out less water in each pump. So we also looked at the rate at which these contractile vacuoles are pumping. And we were kind of surprised that they're really consistent. They're always pumping at around 1 contraction per minute. So they're not changing the rate that they're expelling water. They're only changing the size of the vacuole. And this seemed weird and different from what we know from other systems. So in Dicty, it's been widely reported that if you move Dicty to water, it starts pumping its contractile vacuoles like crazy. So we wanted to directly compare Naegleria and Dicty side by side. So we started a collaboration with Dr. Mark Edwards at Amherst College because he works on Dicty. And using his cells, we found that indeed Dicty is pumping more when it's in water. So more hypoosmotic stress, faster pumping events. And side by side with Naegleria, again, Naegleria is always pumping around 1 pump per minute. So this ability to change your pumping rate is not a conserved strategy between these 2 different amoebae. Next, I looked at the contractile vacuole size. So even just looking at a still frame here, you can see that there are these really large, bright vacuoles when cells are in water. But when they're in sorbitol, you don't see those same really big, really bright vacuoles. So when we quantified the area, we found that just like Naegleria, Dicty is also changing its contractile vacuole size. Okay, so it's not getting, they're not as big when they're under less osmotic stress. So this seems to be conserved, and it's also consistent with what has been reported for Paramecium. So what do contractile vacuoles look like and how do they compare to other protists? Well, their main setup is pretty much the same with this tubular network and large bladder. And like other protists, Naegleria can change its contractile vacuole size, but unlike other protists, it can't change its pumping rate, which gives us some really interesting questions about if Naegleria could be using some type of a molecular clock. And I just want to underscore the importance of looking at these contractile vacuoles, because if you think about Naegleria fowleri, the brain-eating amoeba, it needs to go from a pond water environment to a human brain. And those are 2 completely different osmotic environments. And contractile vacuoles are also something that the amoeba has that human brains don't have. So that could be a really promising drug target for future studies. But for my future studies, I'm really curious about what's driving this ARP2/3 activation, and if it could be scar wave complex, like we would expect if it was a mammalian cell. But to do that, we're developing genetic tools first. So I'm working on getting morpholinos up and running at the moment. I also want to know how these cells are osmoregulating in terms of what specific hormones and myosins could be contributing to vacuole pumping. And I want to know how that rate is held constant and if it could be a molecular clock. And another project I didn't get a chance to tell you about was cytokinesis. So we don't know a whole lot about cytokinesis in Naegleria. We don't even know if it has an actin contractile ring. But when it divides, its contractile network gets completely distributed around the cell. And after division, it goes back to having 1 vacuole for each daughter cell, which is giving me a lot of questions about how organelles are inherited and if actin could be dispersing the network. So with that, I would like to thank everyone in the Fritz Lehland lab, especially Tati, who's been helping me with the contractile vacuole work, and Mark Edwards from Amherst College for the Dicty cells. And I've used almost my entire 25 minutes. So if there's questions we don't have time for, here's all of my contact information. I'm always happy to talk about Naegleria. And now I'll open it up to questions. Thank you.
Great, Katrina, that was fantastic. And I hope Marino Arroyo is watching on YouTube or later on, because he sort of teased us a few weeks ago about sort of our focus on mammalian cells. So I'm glad you're bringing in other kingdoms here. So I had a question to start out with, which is just, can you provide maybe a little bit more context about what your thinking is about why these cells are able to crawl in an adherent way in the first place, because you'd think that they're just swimming around, right? So why are they able to crawl, and how are they doing it, and how is that similar or different than than other creatures, other cell types?
Question. So Naegleria is pretty impressive because it can just crawl on uncoated glass, which a lot of cells can't do. So it's all just got to be like non-specific interactions to give traction to the amoeba crawling forward. I think that the forward protrusions are definitely ARC23 and branched actin mediated. And then, I mean, when it's a flagellate, it can also swim. So it can swim a whole lot faster than it can crawl, even though it does crawl impressively fast at around 120 microns a minute. And actually, if you had really tight adhesions there, that wouldn't work because you couldn't achieve those speeds if you had to disassemble a focal adhesion every time you wanted to move a little bit,
Okay, fascinating. So you guys think it's not really adhesions that maybe someone like me is used to thinking about, but more of a sort of a transient and, like you said, non-specific, sort of just sticking to the substrate. And because I was struck by how similar your movement on your 2D glass looked to the movie that you showed that was in 3D, right? So normally 2D versus 3D for a lot of cell types is like wildly different, but yours, like the morphology was pretty similar, 2D and 3D.
Mm-hmm. Yeah, well, I think that was a really cool finding from that lattice light sheet paper, that even if you're crawling in a 3D environment and you're not templating your protrusions on a glass cover slip, you're still forming these linear lamellar looking protrusions.
Awesome. Okay, thanks a lot for that perspective. We have a raised hand from Margaret Titus. Please go ahead.
Hey, Katrina, really
Hi.
nice work. I have many questions, but first of all, I really have great love for Naegleria because they don't use microtubules. I think that's pretty good. I'm wondering about your contractile vacuoles. Do you did you stain cells with phalloid? Do you see actin around the contractile vacuoles?
Yes, you might have to bear with me on some slides that are after this to find the right one.
And then the other question I have is, did you treat cells with blebistatin? Did that have any impact on contractile vacuole?
Yes, so I have used blebistatin, and blebistatin didn't do anything. But here you can see that there's this really round, kind of actin-coated structure in the cell. And we saw these in some cells, but not every cell. And there were a variety of sizes that were consistent with contractile vacuoles. So I've been really working on optimizing that fix and stain all in one go, because I want to see a vacuole pumping and then fix and stain it to see if there's actin at that structure that was pumping. And I'm close, but not there yet. Okay,
and just one sort of maybe Dick-D nerd question. So you guys are probably using the standard lab strain AX3 or something like that. And it's been shown that strain is mutant in a RAS gap that enables it to do macropinocytosis and live in liquid media. But the wild type strains don't have that mutation. And so they're basically, what I think of as their actin management is very different from the laboratory strains. And I was just wondering if that might have some impact on the contractile vacuole timing and pumping that you're seeing, if the actin management maybe is a little bit different. I don't know if it's linked, but I was just curious to know if you guys had tried to look at those strains.
Yeah, that's a really great question. And actually everything I think every slide I showed you in here was also the xenic strain for Naegleria. So it also is able to macropinocytose crazy amounts. But I've compared the Naegleria xenic strain with the non-xenic strain that eats bacteria. Actually, the non-xenic strain is way easier to look at because it doesn't have macropinosomes all over. If you saw all of the other vacuoles that were in the cell, those were macropinosomes, we think. So the basic pumping mechanism seems to be the same. Actually,
Okay.
that last video I showed, that was the cell dividing, going back to just one contractile vacuole, that was the non-exenic Nuclearia strain. I assume it'd be similar for Dicty, but I could certainly ask Mark for some more cells because looking at Dicty is pretty fun.
Yes, it is. Anyway, really beautiful work.
Thank you.
So is the failure of the Nuclearia to be able to change its vacuole size, is that related to the fact that it doesn't have microtubules compared to the Dixie?
Yeah, I've been wondering about that because the microtubules seem to be really important for Dicty and also for the organization of the vacuoles in Paramecium. So when we don't have any microtubules at all, and like that's the most striking difference that we see between Dicty and Nuclearia, I think it could be likely that that the microtubules are what's allowing it to change its vacuole maximum size. But I think the thing I want to know is how Nuclearia knows to pump every minute, because that seems totally crazy that it can pump once a minute in sorbitol or water. It can tell time.
Yeah, that's really interesting. So we have one last question before we'll move on to Allison from Alberto Ippolito. He says on YouTube, thanks for the great talk, and asks, have you measured the motile response of the cell type on different substrate curvatures?
No, but that sounds really interesting. Yeah, we haven't looked at that at all. I do want to point out that I didn't mention this at all, but when you squish the cells under agarose, it looks like they are blebbing more than making like thin lamellar protrusions, which has also been seen for other things. Like even if you squish coinoflagellates, they'll start blebbing as a mechanism to get away and escape. But I haven't tried different curvatures, and if you want to talk about that later, then that sounds like an interesting question.
Right. Yeah, Alberto, she's got her contact info up, so feel free to shoot Katrina an email. All right, thank you so much. That was really fascinating and lots to think about here, and really cool what the connections are between, I don't know, for me, the cell types I'm more used to looking at. Thanks so much. All right, next up we have Allison Sgro, and go ahead and start sharing your screen whenever you're ready. So welcome to Allison. She's a scientist who's deeply excited about how groups of cells work and think together across different kingdoms of life. So she received her doctorate in chemistry from the University of Washington, and then she next did her postdoc at Princeton, where she investigated the origins of collective behavior in dictyostelium. And so in January 2017, she began her appointment as an assistant professor at Boston University. She's in the biomedical engineering department, and she's also a member of the Biological Design Center. So thank you, Allison, so much for being here, and really excited to hear your talk.
Great. Thanks so much for the lovely introduction. All right, so I am, like I said, really interested in how groups make decisions together. And in particular, my lab is interested in how single cell signaling dynamics are used to coordinate these kinds of multicellular behaviors like migration. So I want to first give you sort of an idea of like, how do I think about these kinds of problems, and then walk you through a sampling of two things we're thinking about right now. But sort of how I think about this and how I got on this whole idea as a whole was I got really fascinated by how groups make decisions. I think we have some really nice ideas on how people do this, right? So if you've got people, you can follow the leader, and some king or dictator can tell you what to do, and that works to some level of efficacy until there's revolution and everyone says, I'm not doing this anymore. We can have group discussions, and this may work well in your group meeting, maybe less so in faculty meetings, even worse off if we're going all the way into politics, but we like to pretend that people can discuss what they're going to do and how they're going to do it next. And then people engage in collective behaviors, right? We've got stadium waves, mosh pits, where you can think about people like physical particles that sort of coordinate and have collective migration, other collective phenomena that they engage in together. And I like to think about cells as doing the same kinds of classes of behaviors. Like in your heart, you've got pacemaker cells that beat together. 1 in 100 cells, or pacemakers help all the cells coordinate their beating together, right? These 1 in 100 cells send out pulses that help everybody else coordinate their contraction, right? If these contractions can't be coordinated, or you have more or less pacemaker cells, your heart doesn't work. I'm sure you've seen tons of beautiful collective behaviors in terms of migrating cell sheets. I'll even show a video today of lovely 2D collective cell migration, where physical models think of these cells as particles is very efficacious in understanding where these behaviors come from. But I'm sort of interested in, are there sort of group discussion kinds of behaviors? We sort of combined a number of these approaches and sort of talk things out and figure, you're doing this, we're going to divide roles. And so I've been trying to figure out, do these kinds of behaviors exist? And diving into different model systems is one of the things I've been enjoying to figure out, can we answer this question? So I really appreciate the opening act for a little bit of introduction on dicty, because that's the first one I'm going to talk about today. Before I go there, I want to sort of think about how do you even start to disentangle this kind of question? And so what's the sort of common approach of the two projects I'm going to talk about? And if we ever run into each other, all the sort of different kinds of cellular questions I try and disentangle. So we like to think about this, right, is that cells have inputs and they need to decide how to behave as part of a group. And our goal as an experimental lab that also works with theory is to figure out how do we experimentally control these inputs to quantify how cells process information to decide on outputs, where motility and collective migration are kind of group output they can be engaging in. So for example, if I think about a feature that I'm interested in is cell location, where it is, how it's moving, I can maybe control that input with surface patterning or scaffolds, and I can use time-lapse microscopy, like all these beautiful videos you've been watching, to figure out where cells are and how they're behaving. But I'm also interested in the idea of cells talking and thinking as inputs we can control and quantify. So intracellular signaling, communication between cells is super important. It sort of underlies a lot of the phenomena I'm going to be focusing on today. We can control that input with techniques like microfluidics. It's a little harder to visualize, so I'm not going to focus on those efforts today, but you can do that using protein biosensors in the environment, as well as other approaches to get nice spatiotemporal readout of how cells are actually communicating and talking to each other to work together. But I also like to think about a lot about intracellular signaling, how cells are thinking about the world around them and using that to coordinate their different parts in order to do something like move or move as a group and coordinate with other cells. So you can control that input with a lot of different techniques. I'm going to talk about some efforts with optogenetics today, chemigenetics, different, even more, if we want to go back in time, there's some level of control there with mutants. And we're able to visualize the outputs of this kind of signaling with things like genetically encoded biosensors or previously even dyes are able to help us read out how these cells behave. So most of the initial work I'm going to be talking about today is done in Dictyostelium, the social amoeba. I think this organism is really lovely. They live in the soil. And when they run out of food, they come together to create these structures. And this is the process I'm really interested in. That's going to be the different focus of my talk. And I want to sort of illustrate what this process looks like to you. So this video on the right side of the screen, what you're seeing here before I hit play is a lawn of bacteria. And on the left side of the screen is a feeding front of amoeba. And what's happening here is in this feeding front, they're consuming the bacteria. And at the back side, they're starting to starve. And as they starve, they turn on this program to begin to communicate and work together. So what you're seeing is the cells are rounding up as they receive an extracellular signal that causes them to contract. And the signal comes in waves between the cells. And that's why you're seeing these sorts of waves of contraction. And the cells chemotax up to what they perceive the source of those waves to be into groups. And in these groups, they start to turn on a development program. About 20% of the cells differentiate into stalk cells that vacuolate and die, to hold up the other 80% of cells, the spore cells, that can be more easily stored for longer periods of time, and then carried to an area with more food. So I think this is a really there's so much rich stuff here. There's developmental questions. There's how do you choose fate questions. There's beautiful cell motility questions, signaling questions. How do you coordinate actin and do single cell migration questions? I think this is a beautiful model system. That's why we're hearing about twice in one set of talks. I'm surprised you haven't heard about it more across all the talks this season, but I think there's also been a few other dicty talks as well. So this whole process is coordinated by a pretty complex signaling network, and we know a ton about it. We have a great idea from decades of painstaking biochemistry, what a lot of the key players are, what a lot of the potential links are. Some of these are going to look very familiar to people who do more mammalian cell work. We've got RAS, all sorts of things here. Maybe you also do signaling and are familiar with PKA. We've got a few unique dicty things here that are really important. There's cyclic AMP receptors on the surface. Cyclic AMP is, I think, maybe the key molecule we're going to talk about a lot today. This is the molecule that these cells use internally they generate this molecule to send to other cells as a signal to relay. So it's one of the few systems where cyclic AMP is a primary messenger between cells and not just a secondary messenger to relay signals within cells. And they also use the cyclic AMP more traditionally as a second messenger to control gene expression and other behaviors through phosphokine PKA. the challenge here in trying to understand this whole approach and how it can be responsible for controlling coordinated signaling and collective migration is that, like in any system, if we zoom in on one of these nodes, there's many more components. Some of the links are sort of more or less well known. And how do we, just think about this as a challenging problem. So I don't want to dive in too much to the modeling efforts we've been doing today. But one of the things I like to do is sort of figure out how can we sort of think about what are the most basic essential components of these networks that are important for driving migration and signaling, and to sort of take a top-down phenomenological modeling approach. So I'm going to tell you a little bit about the essential phenomena that are important for DICTE here. We're going to watch a bunch of videos. I hope we're going to have some fun discussions at later dates as we start to disentangle what could be going on here. But I want to give you an intuition for, again, how are these cells thinking about how to work together and how to engage in collective migration. So I told you that collective organization in DICTE is controlled by cyclic AMP, and that we're able to go ahead and watch cells think about this process. So on the left side of the screen here, we've got fluorescently labeled cells. And on the right side, we have the same cells, but they have a sensor for cyclic AMP. And so red is going to be a low amount of internal cyclic AMP, and yellow is going to be high. And in this case, I'll explain why, where the cells are roughly mirroring what you see between the cells with their own internal responses. Let's go ahead and get that playing. All right, so this is just after collective signaling has started, so about 4 hours into development. And in a low density population, the cells take a little while to start sending each other coordinated waves of signaling molecules. And for a little bit, they don't start to respond. So you see most of the interesting behavior over here with these waves passing through. But as the cells develop, they're also upregulating their motility response. And so you're starting to see the cells move pulsally in this left pane in response to each of the waves. And as there's different competing centers trying to stabilize, it takes them a little bit of time to decide where to go. Eventually, something down here becomes really attractive. And then over here, and so the cells start to stream in. You can, if you're looking in the left one, you can see they sort of take on a very distinctive directed shape. And then they finally start going into this one center here. So something I think is really interesting, there's so many different rich motility behaviors you can see in this one kind of video. And they are also driven, we're thought, by different mechanisms at different times. So that initial sort of coordination is thought to be really dominated by signaling, which is what I'll focus on here. But there's labs that have done a lot of work that show, like, once the cells start to touch and stream, there's other really important phenomena that actually adhesion is really important, and you don't even need some of the signaling we're looking at here. So again, really rich system for those of you interested in collective migration. Can't talk it up enough. So in addition to signaling being really key, one of the things that's really important is actually density. And so I want to show this movie from another lab that really illustrates that phenomena. So if you have a much denser system, and so in this video, dark is going to be high cyclic AMP and light is going to be low cyclic AMP. You don't see anything like what you just saw in my movie where cells take some time to stabilize between different centers. You very rapidly are able to establish a center where all of the cells will stream directly in. All right, and so this whole process is driven by how single cells respond to changes in environmental cyclic AMP levels. So one thing that's really key to understand in the system is how does a single cell respond? Because that's going to allow us to sort of disentangle how does a whole group of cells move together. And so the way this sort of data, we were able to learn about these cells is by doing really careful single cell experiments. So we take a single DICTE cell, just like the ones you saw before, but now looking for a fluorescent readout of cyclic AMP dynamics, we can control the extracellular environment using microfluidics. And we can measure internal level dynamics at the levels of single cells using FRET sensors. And the most basic thing you can do to a cell is give it a pulse of cyclic AMP and ask, how does it respond? And what happens in these cells is that if you give a cell a pulse of cyclic AMP, say something very small at the lower end of the physiological range, like 1 nanomolar, these cells will respond with their own internal pulse and then return to baseline. And these are 3 individual cells here, just to give you an example, show you how little variety there is. But if you go up to, say, the top of the physiological range, say 10 micromolar, so this is thought to exist in the aggregates, these cells will spike and then oscillate. Which this tells us these cells are individually excitable. And that's actually really key for reproducing the group dynamics you see. I don't want to dive into too much of published work. There's lots of nice thoughts on this, including some theory work we're hoping to post shortly. But basically, what you're seeing in those movies is that low density populations have synchronized pulses, of those excitable pulses that are synchronized but have trouble coordinating them to get started on that aggregation process. And high density populations, you can see those synchronized waves passing through. Then the key parameters that control the frequency of those waves and the ability to even begin collectively migrating are cell density and degradation rate, which you control via flow. There's, again, a lot of beautiful modeling work to understand how this originates that you can check out. But what I wanted to do is to get you in this idea that these cells are single excitable cells that respond with pulses, and they expect pulses in the environment in order to chemotax toward, to help you understand some of the data I want to show you about controlling collective cell migration. So one of the things we've been trying to understand is what's important to these cells, and we're taking a proof by control approach. So the idea here is you stimulate a single cell, turn on cyclic AMP generation, then they're going to use that again to start coordinating internal programs like gene expression, but also send this to other cells to coordinate group motility behaviors. All right, so the idea is here you can induce aggregation and begin to understand the rules that drive this whole process. It seemed like a really great idea when I got started. It's been a sticky rabbit hole, but I think there is some fun stuff in here. And I want to sort of show you some of the beginning things we've been able to figure out and what the sort of next steps are. So to dig in a little bit more deeply, we're able to modulate cyclic AMP production using a blue light activated adenyl cyclase. It's been used in a few other systems. Particularly, it's one called BPAC. There's a few different approaches to controlling cyclic AMP production, but this is the tool of choice for a number of labs because it has two really key features. The first is that it has almost no direct activity. The dirty secret of optogenetics is that a lot of these tools are often leaky for light, and DICT are very sensitive to cyclic AMP. So if you use other tools, it just creates a ton of cyclic AMP all the time, and you can't really modulate the levels at a level that's appropriate for early development. It can be really useful for later development, but not at this early stage where they're only using a little bit of cyclic AMP. And also has a pretty high dynamic range, 300 to 500-fold exposure on increased activity to blue light. So just to give you an example of how this works, if I put two cells next to each other, one that has a fluorescent sensor and one that contains the BPAC, and I excite and image them separately. If I'm imaging the dynamics of internal cyclic AMP inside this sensor cell, and then I start activating the other cell very close by at 2 minutes, this sensor cell will receive cyclic AMP from that cell, respond with its own pulse. All right, and so we're able to use BPAC to control collective signaling through sending signals to other cells, and then we can go ahead and use that to control collective migration. So something I think is really just amazing. I talked about how I really find signaling and how groups make decisions together super interesting. And then in the heart, you have 1 in 100 cells as a pacemaker cell setting the pulse. So here is one in the center. I'm going to hold my mouse on it. Is one BPAC cell, and I'm pulsing it for 3 minutes on, 4 minutes off. That's about the frequency of those natural waves you see. And one cell is sufficient to drive collective migration into this cell in the whole group. You can see that the group does try and coordinate other signaling centers, but one is enough to take over everybody's dynamics. I just find this really remarkable. And so it got me thinking about what else could be important and what else can we learn? The answer is it's a little bit thorny. It's not super obvious. I want to show you some cool movies, but there's still a lot of work to be done here. If you think about it, there's a bunch of parameters that we can control experimentally that could be important. They could be the number of cells. So I already showed you how density is important for that sort of initial decision on how do we even get started to signal, how much they release, where they're located, how you stimulate them, the duration and timing of stimulus. I want to give you a spoiler is that aggregation and collective motility in the system is incredibly robust, and that shouldn't be a surprise on some level. These are microbes that are live in the environment. In these videos, I'll show you some other videos that use non-lab strains. Even there, right, their basic fundamental design principles of how do we work together are meant to work over orders of magnitude different changes in group size, really in variable environmental conditions. That's something I think is really cool about microbes. They have incredibly robust programs to deal with an incredibly robust, so they continue to be robust in very different environmental situations. Something I think is particularly neat is that continuous stimulation doesn't really drive aggregation. It's sort of, if we put a few control cells around, say one here, and continuously stimulate, you might expect like, oh, it's like a point source of cyclic AMP, cells should go toward it. And it doesn't really drive everyone in the same way. We get these other competing centers that start to win out and that this cell ends up in other centers. And something I think is really interesting, you might expect it would be different if you could just put a needle and have everyone come straight in, but it turns out it doesn't work. The cells really like to see a repetitive stimulus. Something else I think is cool is that even if you stimulate too slow, much slower than the natural frequency, you're still able to compete with other natural signaling centers. So we have a few red cells crawling along here. I fear this is one of the things that doesn't work super well on YouTube, so I'm going to try to highlight with my mouse. But we've got a few of them right here in this signaling center, seating this one up here, and one down here. And it turns out that they each become pretty attractive and then these centers merge together in this structure. And so they're able to outcompete the other naturalizing centers, even when they're stimulating too slow. But something I find really interesting is that too fast doesn't seed centers. I mentioned that these cells are excitable. I don't want to go dig too much down this rabbit hole, but there's also a refractory period. So if you don't give them sufficient rest time, they're not able to propagate waves across a population. And these 3 cells here are not really able to seed centers. They get a little bit of cells near them, but they're really driving too fast. And they end up going toward other densities of cells. Up here is really important. And finally, this one up here stabilizes. Not down here, we had these original cells. One of the things I think is really cool that we can do is that we can actually induce the other collective cell migration phenomena. So we put a bunch of these cells here that we can control and turn up the light really high. We can actually induce a collective migration state known as a slug. This is a later part of dicty development that sometimes happens, where I told you these cells exist in soil. Sometimes they need to crawl out of the soil to be up the surface. And I mentioned a lot that higher levels of cyclic AMP are used later in development. And so if you provide these higher levels of cyclic AMP, you can actually induce the whole group of cells to crawl away. So I think that's a really neat place where we've stumbled upon a key design parameter of how this whole system works by modulating levels as well as oscillatory dynamics through time. So where is this going? I've shown you a lot of videos and you're thinking, well, that's cool, but like, what can I learn? The challenges here is how do you stimulate and control? A lot of these fluorescent tools don't work well in conjunction with optogenetic tools because they use the same frequencies of light. And so we finally hit on a nice sensor and an ability to be able to visualize and control simultaneously in the same cell. So this is work done by Dr. Chiara Ricky-Tam and Chinwen Zhu. So if we go ahead and we're able to watch here, every 5 minutes, we're able to stimulate these cells, and they're going to go dark when stimulated with the same tool. And that's sort of new development we've gotten working during the pandemic, where now we can go ahead and, as lab things ramp back up, take a bunch of data to sort of now understand what's the relationship between these waves, all right, and what are our cells sending out? How is it competing with the natural population? And then how does that control this whole collective motility process? Because that's the key piece that's been missing in the data I've shown you so far. That's really hampered our understanding of how do all of these components work together, right? What's important here is we're trying to compete with the natural process. And so if we can't see the natural process we're both competing with and creating, we're not really able to disentangle everything. It's not quite so obvious. Something else that I think is really important that was hinted at in the previous talk was natural isolates. So they have, as was mentioned in the question session, a very different way of coordinating some motility. And they also have, in fact, very different characteristic signaling patterns and frequencies, both frequencies of signaling and also spatial patterns they create. This is through potentially tuning both signaling dynamics, but also things like adhesion between cells. So you can see here, this is a lab strain on the left. This is a natural isolate strain that feeds on bacteria on the right. If you're imagining how the signaling dynamics are different, they are. The natural isolate is actually a little bit slower, but they have very different timing of when they start to segregate, the spatial patterns they create. These natural isolates are somewhat genetically tractable. I will tell you that they're pretty amenable to putting in single color fluorescent proteins. We have that working pretty well. I'm going to show you some of that data here. They don't really like bigger genes for working on this. Similarly, work where we've been able to put in a green fluorescent protein for natural isolates show that actually their partitioning is incredibly reproducible, as you can see here on the left. You can, if you plate the cells reproducibly, they will in fact reproduce the same spatial pattern in terms of partitioning every time. But I also want to call your attention to something on the right, where the cells sort of pre-aggregate, dissociate, and then come together in bigger aggregation groups. We're really excited about this sort of not chemoattractant, but chemorepulsive behavior that seems to be important in early development. There's been a few reports of this in other systems and also in dicty at later times, but not in this early stage. So something else we're really interested in terms of collective motility. All right, so I think I'm running a little bit short on time. I want to quickly preview and talk about a project I want to basically just have discussions with you about. This is totally a new thing we got started during the pandemic. Probably because I got really excited about staring at a lot of mammalian cell videos that are so beautiful in these kinds of talks. But we're really interested in understanding, again, how does a single cell signaling dynamics coordinate group behaviors like collective migration, but here at the level of how do cells coordinate protrusion dynamics. So just to give you a quick preview, hopefully just to spur further conversations, I'm sure I have missed your favorite paper or probably all of your favorite papers and all of the, I really want to hear about how we're wrong about this. So please reach out to me and tell me afterward. But we've been putting together a proposed network for how PKA modulates protrusion migration dynamics through ROA and RAC1. And just to quickly sort of summarize and give you the ability to come have discussions with me about this a la a conference, we have the ability to control spatial temporal TPK dynamics through cyclic AMP signaling using the same tool that we use in DICTI. Those of you who also may take an optogenic based approach might notice that we also have optogenic control over some other components in this network that allow us to test this model. And so we're working on slowly getting all of these components up and running. But in particular, PK is a great place to start modulating because these tools have fast on-off kinetics, high sensitivity, and low phototoxicity. And what's nice is that since there's been a wealth of literature, we're able to put together this proposed network, and we combine math modeling and experimental approaches to test this. So we have a sort of, I don't want to run you through all the math on a YouTube talk, but we do have a model set up for how this works. And we can use it to reproduce two key experimental observations we think are important. The first is oscillatory rack and row A dynamics. And the second is that after you activate pKa and you see delayed and diminished membrane dynamics and less protrusion formation, if you take away the activation of pKa, these dynamics are recoverable and cells can recover. And so just very quickly, again, because I know I'm at the end of my time, we are able to actually reproduce these oscillatory dynamics, and as well as see these both delay of membrane dynamics and then recoverable dynamics if we add noise to this network, where we can turn off the oscillations and then recover them as we add noise into the system. And so where are we going with this? Just to go back to the video I showed you, what this was is a bunch of NIH3K3 cells. We've labeled F-tractin. You can also do membrane markers, and we're able to go ahead and stimulate and see how do the cells respond? How does this control the protrusion dynamics? And so that's where we're digging in now, as well as really diving in to understand how this model behaves, where the oscillations come from, how sensitive to parameters is it. So I'm excited to maybe have conversations with you about this. And with that, I want to thank the team that did all of this hard work. And in particular, again, call out people focusing on dictyostelium. And I'll take questions.
Great, fantastic. Thanks, Allison. Folks, as usual, you can leave questions in the chat or use the raise hand function. One question that I had that's a little bit, I don't know, maybe this is silly, but I was wondering about the cells that are left behind. I noticed when you have the aggregation, there's always, I don't know, 5%, something like that, a small number of them. And I would think there'd be some evolutionary pressure to not be left behind. I mean, are those interesting at all, or
I couldn't have planned a better question myself. I swear I didn't. So I had this same discussion with a collaborator, Karina Ternida at Princeton, close to a decade ago. And we have a few papers on this, that these cells that are left behind are viable. And they're actually really great in that. For a long time, there was some confusion about, well, if there are natural isolates, in particular, that are better at getting to the spore or the stalk, you've got clear winners at this. Like, how can you have things that are bad at becoming stalk cells? And the answer is these cells that are left behind are viable, and they represent an opportunity to take advantage of when nutrients returns to this area instead of going off to somewhere else. And so that you can sort of bet on different strategies. So yes, they are viable. The other way to think about this is that they could, you can just think about development being a probability distribution of like, well, some things have turned everything on, but some of them are slower to turn everything on. And if you do actually wait, and you can see this in one of the earlier videos, that some cells aggregate initially, and then later you'll see other groups of cells aggregate later. Those are those same cells that are left behind. A lot of these videos are 24 hours. And so it is true, if you leave the video running, take data for 3 days, you'll see these other cells also potentially aggregate as well, but getting 3 days of microscope time on one experiment is a lot of, doesn't happen unless it's a really empty weekend.
Okay, cool. Yeah, well, I had no idea that you guys had already looked at that. And it's interesting that maybe they're just a little bit slower, right? The cells that are left behind. And then also that last movie that you showed under the future directions heading, the natural isolates, that was just stunning watching them sort of aggregate up and then swim away. Was there any reason that they were all swimming off to the left?
Yeah, so these slugs are phototactic as a group, and there's a lot of, again, really interesting data here where there's mixed evidence that the single cells themselves are phototactic, but actually the group as a whole is incredibly phototactic when they're in that slug state. And again, you can imagine it's something that's been highly selected for that. If you're aggregating in the soil and you get a pinpoint of light coming through, I mean, they're also thermotactic as well, right? So you want to get up there, right? So they're really attuned for that. I assume what's going on in that video, we didn't have this recorded in that. It's a it was taken in a big chamber with fluorescent lights coming down from the sides, sort of a la a black stage theater, a black box theater stage. And I imagine that it's a little bit angled, and there's a slight light gradient that isn't obvious in the video. And then, of course, by the time we got the data, it wasn't quite clear. But yeah, they're incredibly sensitive to that.
So they are responding to something. It's just not totally clear what it is.
Yes, something we'd like to do.
Fantastic.
But you can definitely get them to phototax the small gradients of light. So we think that's what it is.
Okay, great. Yeah, we'll wait a minute or two here. Margaret, go ahead. I had a feeling you'd have a question.
Yeah, beautiful work. That's really great. I really like the natural isolates because, as you might have gathered, we're playing with them a little bit, and they're fabulous. They're really great. Their motility is wonderful, and they make beautiful phylopodia, so we're really excited about them. I had a question about your cyclic AMP, the optogenetics that you were doing, because I'm just wondering, how much control do you have over how much cyclic AMP you're producing? And so how much can you scan, the sensitivity of that? Because it's also a consequence of how much the cell is producing and then how much it's able to export out. And so I'm wondering, how much fine-tuned control do you have over that?
We do have a good amount of fine-tuned control. We don't have a great amount of readout of it. Once it is very sensitive, so you're getting down into using a lot of ND filters to block out light, right, and tuning your source way down. But you can actually tune it down so you see some simulation, but not as much, and tune it way up so you're maxing everything out. The challenge is, the other issue is, how we can get reasonably uniform expression, but of course there's some variability, so it's hard to read out what's the variability there, and we can't get an absolute level. And again, there isn't a great, there's a medium understanding of how things are exported to dictyostelium, but it's not clear how regulated that process is at certain times. So basically the answer that we're trying to take is that once we get that sensor in, we can at least see what it's getting from another cell and use that in tandem. I think that's going to be the key approach, but you're right. We can't put a number on it. If we put in, 2% LED power, we are generating 500 nanomolar cyclic AMP in a cell. But it is definitely tunable, and you can tell that from the response dynamics across experiments.
It's really cool. Beautiful work.
Thank you.
Can you, I'll have Jonathan Chubb ask his question first. I have kind of a methodological question of my own, but Jonathan, go ahead.
Hello. How are you doing? How long do you have to stimulate for to create a leader in the aggregation experiments? Is it just a few pulses, or do you have to go all the way through?
That's a great question. I didn't put these videos in. It actually has to be pretty persistent throughout. You can start very late in development. So, I usually shoot for between about 3/4 of a monolayer, and at that density in terms of timing. If I don't start until 7 to 8 hours in, it doesn't matter. No early dynamics don't matter. Again, at that density. If I went higher, I think it would matter earlier. But if I start then, it doesn't matter. But I have to be persistent after that, and persistence is really key. I haven't been able to speed it by doing it for 2 or 3 cycles. That is not, that hasn't worked. Basically, I thought maybe if there was like a key hour, if I turned it on for this key hour or key 2 hours. I think it is partly the issue is that, and I think it's partly I should just up density. I think the whole thing goes a lot faster, and there is a really key time. And if I could triangulate down to, it is always between hours 6 and 7. And if I do hours 6 and 7 after this density, it might work. That's my sort of, there does seem to be indicators there's a key commitment time, but it isn't such that like, if I have any variability in my density in what seems to be a not very broad range of density, it's still, the timing is widely variable. So that's been the challenge. I hope that's going to be mitigated by going up to a much higher density.
Thank you very much. That's cool.
I'm wondering, so are these cultures, are they in liquid media?
No. So everything you've seen is on, there's 2 ways that these are done. Most of the ones that I've shown you are on glass with an agar overlay. So that's why those groups break up, or they don't always crawl away. A few of the videos with natural isolates are done on agar from the top down. But they want a sort of moist environment. I do find you can get them to aggregate in water or a light buffer. I personally find that they tend to get sticky first, and the movement dynamics are a little weird. If they're just completely ensconced by water, so this humid environment, again, a la soil, it seems to be really important.
Okay, because yeah, so I mean, are there signals that are transferring through the environment at all, or sort of akin to like a soluble signal that I might
Yes.
think of like
So
in
they're
the
sending
body, or is everything cell to cell?
Yeah, so everything's going, there is a diffusible signal, cyclic AMP. It does work best when it's really in close proximity, but you can imagine, again, if you have this third dimension, it's diffusing away in the third dimension, and that's not super helpful. We do take advantage of that for some of the single cell signaling experiments, but in the group experiments I've shown you in the videos, those are all done without that because we find that really appears to perturb the dynamics.
So it's extracellular, but it's not in a medium. It's just sort of locally on the cells or something. Is that
it is diffusible between cells? It is meant to diffuse,
okay.
But the issue is that it rapidly drops off to not. You need a pretty big pulse, it appears, and so if you have too much distance between the cells, you don't have great signal relay.
Right. So that's that density parameter that you're saying that you need to have a minimum density.
Yeah. I mean, over
Okay.
time, what'll happen is they'll get closer together and that'll help alleviate the situation.
Right. You have some random movement and then critical density just happens at times. And then those cells can become a signaling center.
2 to 3 days, but it will happen.
Okay. Very cool. Olivier Pertz, go ahead.
Yes, hello. Thank you very much. Fantastic movies. I'm totally jealous. Your cells do in 20 minutes what my mammalian cells take 24 hours to do. So you will have a much higher experimental throughput than we have. So I mean, all the responses about activating cyclic AMP in a subset of the population of cells, I don't know that system, but I could not grab how you get this patterning by intuition. So do you have predictive model that explain you how you you control a small amount of agents of the population, and this somehow feeds and the whole population doing something? I mean, I don't understand it. So you understand it by intuition, or do you understand it by models? Do you have that?
There is a lot of nice modeling work upon this that you can get both stochastically driven centers that can create these waves. I can send you some links afterward to some of the work there on this, but yeah, there are spatial models that show that how you can tune different types of waves you see that will seed these centers. I think our spatial understanding is less well developed than our temporal understanding is, but yes, it is not just intuition.
And then what can you learn from manipulating a small subset of agents in the population? How can you, what does this bring something in you to the models, or?
What I'm hoping it'll bring is that there's, this is sort of hinted at in the work that we have in prep, and I hope will go up on the archive this month or early next month. But, is noise really key for starting and maintaining these behaviors? And so actually, I'm going to totally confess, part of what I wanted to do with these cells is try and see how important having stochastic signaling behavior is, and how are different frequencies of noise or levels of noise really important or not, since it's one of these key model predictions about what's really important for maintaining and creating and maintaining group behaviors. So there's, again, several different ways you can get it in models. There's experimental data that hints that both are important. And so that was actually my original goal, is I'd like to use that to disentangle that. So that's where I'm going. And so in that sense, like, can we learn something about nature? I hope that's something we can learn about, is how important noise is and how important it is for tuning different behaviors we see.
Then maybe a last question. In the RACRO model in the fibroblasts, so the network that you've built, it's how do you get the parameters, and are the simulations, are they robust against the parameter space?
Some of them we can get a little bit of hint from the literature, and they are a little bit parameter space sensitive. We're trying to we're basically still in development. They are not so specific that one needs an exact specific value to get them. It is medium robust, and we're trying to basically now in this sort of digging in to make sure it is more robust than just we happen to pick the right one. So I agree with the underlying question there, which is this a model that's only going to work for one set of parameters? It does, there is a little bit of tuning required, but hopefully not a ton. And so my attitude on this one, if it only works at one parameter, then we have the wrong model. And so we are digging into that. But this is, basically the very first set of simulations and results. So it appears looks good so far, but we'll find out. And Junk thinks if it is too sensitive.
Thank you.
I think there's a question in the chat. Natural frequency that I mentioned, how is this determined? I use this term in terms of like a natural, what's the frequency that the cells end up oscillating at as they're aggregating? I basically just, plot, I usually do a wavelet transform of like, what's happening through space and time, how are these cells, what's the sort of intrinsic oscillation frequency? I find that there's a, other people have found this as well, that the cells level off at a maximum frequency. And that's what I think of as that each strain has a different characteristic natural frequency instead of natural behaviors. And again, I do find that if you plate different natural isolates reproducibly, you get a signaling fingerprint that appears to be very reproducible. So they do have a, programmed in pattern. If you are reproducible, they are reproducible, but they can reproducibly recreate the phenomena even if you're not reproducible. And so that's, I think, a really, again, very cool feature of microbes.
I completely agree. The robustness in the system is really incredible, right? We would struggle as engineers to design systems that are so robust. But nature has done it. All right, fantastic. If there aren't any more questions, then I'll go ahead and wrap up for today. So thank you so much to both Katrina and Allison for fantastic talks. And we will see you guys next week. So next week is the last one that we have scheduled for now, and we're still figuring out what things look like in the summer and the fall. So that'll be forthcoming. And with that, I'm just gonna stop the YouTube. So